Okie dokie. So now let's continue on talking about how enzymes do their thing to lower this activation energy barrier, delta G double dagger. Okay. And so this big protein is organizing the reactive groups provided by either a prosthetic group, a coenzyme, perhaps a metal, or by functional groups of the side chains of the amino acid residues into close proximity with and in the proper three-dimensional orientation to facilitate this reaction and lead to this transformation. Okay, we're going to see that the binding energy helps drive this to lower this activation energy barrier, and we're going to be forming this Michaelis complex. Remember, it's this complex resulting from intermolecular interactions between the enzyme E and the substrate S. So this is the Michaelis complex. Of course, when you bring two things together to bind, we're going to have an entropic penalty. But the take-home point is that, as we'll see, the enzyme binds to the transition state best, okay? And that lowers the activation energy barrier to allow the enzyme to do its thing. How does it do this? Well, the active site of the enzyme is complementary to the transition state of the reaction, okay? The transition states will bind better than the substrates to the enzyme, okay? If it binds better, that means that there are additional or stronger interactions with the transition state as compared to the substrate. And that lowers the activation energy barrier. This because we're talking about intermolecular forces forming. This is largely an enthalpic contribution to the activation energy barrier. So delta H double dagger. So what am I talking about? So let's consider an enzyme. Well, let's consider a reaction, okay? We have a stick, this rod here, and we want to break it, okay? So if you're going from a stick and breaking it into two pieces, you snap that in half. That bent stick, before it breaks into the two products, that bent state, the bent stick is the transition state, okay? So we look at our reaction coordinate, woo, super high barrier. The apex corresponds to the transition state. Okay. Now let's consider an enzyme whose binding site is complementary to the substrate. Okay. Let's consider the substrate to be a metal stick, so magnets will bind to it in this pretend example. If that enzyme's active site is complementary completely to the substrate shape, okay, we form this Michaelis complex here, but there's no driving force to get it into the product because this enzyme substrate complex is super stable because it is so complementary in its binding. If we look at the reaction coordinate diagram, what you can see is this complex is so stable, even more stable. So to overcome and break it to the bent stick, we have an even greater activation energy barrier to overcome. So this is crap. Now, let's see what happens when our enzyme with the active site of magnets is complementary in shape to the transition state, okay? So we have our substrate, the metal rod, binding. It doesn't bind very well. There's only very few intermolecular contacts, okay? But we get more interaction when the stick is bent before it breaks, okay? So this, lots of intermolecular interactions between the transition state and the enzyme, as you can see here in the middle part of this reaction. And this eventually leads to the formation of broken stick P products, okay? Again, if we look at the reaction diagram, we form this enzyme substrate complex. Not so favorable. But it's not as stable as this middle one, right? Okay. And then we overcome to form the product. So this is what we mean by the enzyme is complementary, the active site is complementary to the transition state. So, hey, is there any experimental evidence to support this for a discrete case? We can plot the reaction velocity, which we'll be talking about in the next video. Plot this as a function of substrate concentration. And what you can see is the rate of turnover into product quickly saturates to a maximum velocity. That reaction will only go as fast as, it won't just be infinitely increasing. There's a saturation. It asymptotically approaches a limit. That is related to the amount of enzyme that is present. So there will be a limit, as we'll see, to the amount of complex that can be formed. This will become clearer again in our discussion when we talk about the robust Michaelis-Menten model. But basically, what we can see is that the reaction velocity at low concentrations is proportional to the substrate, and then it will reach this limit, okay? There also is experimental evidence supporting a discrete Michaelis complex from X-ray structures. And you may say, hey, hang on, Sheets. X-ray structures, you said, were not dynamic. How can you measure this kinetic? Kinetics are dynamic. What the heck's going on? One can diffuse in a transition state analog to basically trap it. You know, the analog is not competent for the reaction to proceed, but it is complementary and to the active site conformation. So it basically traps it. And so you can determine structural information that way. Or you can monitor some type of spectroscopic change as a function of the Michaelis complex forming. So typically an active site is not on the surface. It's in a protected environment. It might be a deep cleft or crevice for this protein. This restricts the solvent that might have a deleterious effect on the reaction. It also protects the environment if there is kind of an unstable intermediate that's formed. There's some protection there. By having this cleft or crevice and where the active site is found, this also is kind of changes the dielectric field of the amino acid residues in there, and that can shift the activity, the effective chemical activity of the residues in that active site, and can shift the pKa's. So it's kind of a special, can be a special environment. But proteins are really quite large. Many substrates are often small molecules. So why do you need this honking big protein when it's acting on and transforming a tiny, relatively small substrate? Well, that three-dimensional structure helps hold the residues in place so that they're in the appropriate three-dimensional orientation to facilitate this chemical transformation. You can have a scaffolding or regulatory sites that might be quite distant. For example, allosteric upregulation and downregulation of where the effectors bind. You can channel some reaction to the next fast reaction. And we have these unique microenvironments, as I mentioned, just to illustrate how, even though in the tertiary structure, you see these highlighted stick models of specific side chains of residues that are important in the activity of lysozyme. So they're close in space three-dimensionally in the tertiary structure. If we look at the primary sequence, it's shown here schematically, you can see that those key active site residues are actually quite distant when we compare the actual sequence. So the substrate will bind with weak intermolecular forces. So again, when we're talking about intermolecular forces, we're talking about dipole-dipole, hydrogen bonding, salt bridges, van der Waals interactions, okay? And the active site is complementary, binds to the transition state best. What do we mean by this? Though, because of that complementarity with the transition state, we're forming all these beautiful intermolecular forces that is very enthalpic. So the enthalpic contribution, the delta H double dagger contribution to the activation energy is exothermic, okay? We can think about the binding with respect to two models that we encountered first in the preceding chapter with respect to hemoglobin, the lock and key model as well as the induced fit, okay? And so just as a review, you have preformed binding site and a substrate. They fit together as lock and key, very complementary. And we also saw induced fit where the enzyme, one partner changes its conformation in response to something binding to it. So substrate doesn't change, but it affects a change upon binding, changing the conformation. Vice versa, you could have the substrate changing its conformation in the presence of a more rigid enzyme, or both the enzyme and the substrate or ligand can change their conformation. This is induced fit. We saw several examples about that in our discussion about hemoglobin. So how do enzymes actually lower the activation energy? This is the enthalpic contribution to the free energy of the transition state, okay, this activation energy barrier. Remember, any free energy has an enthalpic contribution as well as an entropic contribution, okay? Since we're talking about delta G double dagger, we're talking about enthalpy double dagger and entropy double dagger for their respective contributions to this. If we are forming these beautiful intermolecular forces, this is exothermic, okay? There are more contacts. So this is an enthalpic contribution to lowering the activation energy barrier that offsets that entropic penalty of bringing these two things together, okay? However, this is a transition state. By nature and by definition, this is unstable. So we're here at the top of the peak, at the transition state. It can either collapse back to substrate, or at the top of the mountain, it could collapse to form product, okay? The transition state is unstable, okay? So in the next little video, we're gonna start talking about how do we talk about the kinetics mathematically and what are some models that will describe this.